Yes, I think it's a pleasure for me to be back in Bologna, although virtually, and present you an overview of this topic. So my idea in drafting this talk behind the more or less flashy title about nanoscale pharmacology and biochemistry is to try to provide you, either as a specialists or non-specialists, as I presume most of you are, with an idea of how you can use fluorescence microscopy to make some quantitative statements about, let's say, biochemical and, let's say, cell biology questions. So my idea will be to start, first of all, to move to the next slide, which is non-trivial, apparently. Okay, yes. So I cannot, yes, now I can do it. So to start with a quick discussion about optical microscopy and fluorescence labeling, then hitting the diffraction limit and breaking the diffraction limit, and then move to how to use the notion of small fluorescence fluctuation out of equilibrium, which is a more physical thing, so I didn't lose completely my roots, to extract information about dynamics of molecules within living cells. Then I will try to give you a very quick and superficial, but I hope at least stimulating overview of how to capture diffusion in space and time. And then I will go to an application. Given the time constraints, I have reduced a little bit the scope given compared to my more ambitious abstract that you might have found on the website, but I will still give a hint to all the application I listed. And of course, if there are questions, feel free to interrupt or raise a hand, or I don't know, find a way in the Zoom setting to ask. So, about optical microscopy. So microscopy in general, and this shouldn't be a surprise to you, is basically the attempt of humans to basically expand the resolving power of their eye. And optical microscopy, which includes fluorescence microscopy, was actually one of the first steps in this direction, which brought the ability to resolve objects, let's say, below 1 micrometer in size. However, over the last century, there have been definitely even more powerful steps towards increased spatial resolution. And one of them is the electron microscopy, which is a bit over a century old as a method, which brings you down to the scale of 1 billionth of a meter, 1 nanometer, and also scanning probe methods, which also allow you basically to go down to atomic resolution. And this is where I want to start from, just to tell you what we are capable of, so where we can start off in terms of resolving things. So scanning probe microscopes are basically nanoscale tips or nanoscale fingers, which you can slide on a surface. And given the nanoscale size of the probe, they can resolve nanoscale features. And this is a very stunning picture of the surface of silicon 111, where you can basically see that each of these dots correspond to an individual atom. So this was a conceptually new microscopic tool, which was introduced basically over almost 35 years ago. And so optical microscopy, on the other hand, relies on an instrument, on equipment, and also on physical principles, which are the same since its invention. So the microscope of today looks very much similar in concept to one of the early microscopes from a century ago. Well, the first microscopes date back to the 17th century, but actually what you can image with this microscope, if you may remember, or at least this was my initial experience in high school, where actually I was using microscope not much different from this one, and I have to say I was not really impressed, are these. So you can look at living cells, but what you can see of these living cells, at least at first sight, is not so tremendously exciting, because you can see the feature of these cells, you can detect that there is some morphology and some, let's say, finer texture to the cells. You can, for example, identify the nuclei, and these grainy features here, but which are the organelles within the cell, but the ability to make quantitative statement with an optical microscope, let's say with this type of approach, is limited. So, of course, I'm deliberately dismissive just to show you where we can get. So things improve or can change drastically the moment you have the possibility of selectively labeling or staining what you're interested in. And there are a number of biochemical and biochemical strategies to, for example, selectively label like the membrane of the cells or the nucleus, which we see here, or let's say features like the cytoskeleton or specific organelles. And one of the most powerful way to do this, which I would like to present as one of the main revolutions which allowed optical microscopy to become so popular over the last 30 years, is the use of fluorescence, and in particular, endogenous fluorescence. So it was discovered not so long ago that some organisms like jellyfish express a protein which is fluorescent. So fluorescence is the mechanism by which you throw at a molecule some light, and this molecule answers with light of a slightly redder color. So in this case, the green fluorescent protein, you can heat it with the blue light and it will answer with green light. So the advantage of a protein is that a protein can be basically genetically encoded. So a protein can be produced by a living organism provided that you provide the genetic sequence for it, and which is represented by this DNA plasmid here. And the other advantage thing about proteins is that you can glue them together by gluing their genetic sequences one after the other. So which means that since cells are mostly made of water and proteins and DNA, so let's call it nucleic acids to be more general also, because it's not just RNA, there are also RNAs, as we saw not so long ago. So you can feed the cells the genetic sequence for your protein of interest, fuse to this kind of glowing bag, which is the green fluorescent protein, and make cells glow. So this was the first demonstration in the mid-90s of the visualization of bacteria, which are excited by blue light, and the bacteria on the right express this green fluorescent protein, and they're bright. Of course, this was the first proof of principle, but then people started to devise all possible ways to label interesting things fluorescently. So from individual cells, developing organism, features within cells, to entire animals or insects in this case. So this approach has also the advantage that you outsource to the cell all the work, because you just give her the genetic instruction to produce your fluorescent label. And so with a fluorescent label in your hands, you can then start to, one moment. So of course, this advancement was so fundamental that it was recognized in 2008 with the Nobel Prize for Chemistry. And you see here the three recipients, with Osamu Shimomura, who was the scientist who basically extracted the green fluorescent protein from the jellyfish, Martin Chalfie, who was the first to actually feed the gene of the GFP to living cells, making them glow, and Roger Chen, who was instrumental in the development of a number of, let's say, tools and the developments of these fluorescent proteins and sensors to actually address meaningful biological questions. So with this in our hands, then we can start looking at a cell with a specific labeling. And what you see here is a fibroblast, is a specific type of cell. And what you see is labeled, so it's being now stained and is giving a contrast, is not just the thickness of the cell, as in the fried egg image I showed you at the beginning, but it's actually the cytoskeleton. In this case, we see actin, which forms basically the structure which keeps the cell in shape. And with its fibers in this region, or its accumulation here at the leading edge as the cell is growing and it's polymerizing to allow the cells to protrude and move. So in principle, then we have the ability to look into a living organism where we have labeled our targets of interest with, let's say, fluorescent proteins, and see what happens. I start talk showing you the visualization of individual atoms. So one legitimate question would be, well, can we now zoom in here and start to see individual molecules? And in order to do so, you can zoom on a small region here. Here the scale is 250 nanometers, so 250 over a billion of a meter. Fluorescent molecules which are emitting light at a certain distances, and then you bring them progressively one towards the other, there will be a distance where the two things will become indistinguishable. And this distance is exactly in this range, which let's say is about a quarter of a micrometer. So that's bad, because then we say, okay, so fluorescence is nice because it allows you specific labeling, but then our resolution is poor compared, for example, to electron microscopes or atomic force microscopes, which I showed you earlier. There are solutions. So one solution would be to make individual molecule stand out. Because if a single molecule stands out, you know that basically is a circular or a spherical emitter. So you could basically say, well, I now see this molecule, which is different from the background. So maybe, I don't know if this will work now in this setting, but would anybody have an idea of what would be a practical way to achieve this kind of making molecules stand out compared to the other molecules present in the sample? Maybe, I don't know if people want to use the chat, or maybe I don't know if we can make this a bit interactive, but or even if there is a chat. But okay, so maybe yeah, we leave this more interaction at the end for the question. So one way, of course,
there is an answer.
There is, because I cannot see. So
you can see the chat line.
Wait, it's now because
Francesco Formaggio says endogenous signals.
Yeah, well, right. But your question now is that you have all molecules which are emitting in the same, so which basically all look the same. It's like you look at a square of people which all wear the same hat. Let's say a green hat. And then if you look from a helicopter or a drone, to be more up to the time, and you look down, you basically just see a green homogeneous distribution. So the question is, how can you? So if molecules were people, you could tell, well, now somebody please wear a red hat, and then you could pinpoint this person. But molecules, you cannot interact like this with molecules. So the question is, yes, you could, for example. Yeah, so you could do things. There is a comment from Marco Marcia in the chat. You could say, you could, for example, tell some people in a region of the square to leave, so to keep this analogy, and then let people one at a time come back in this empty region. So like the police comes, pushes everybody away, and just a few people stay inside, the people and what they do. So this will be one way. Another way will be to color things differently. For example, making people wear hats of different colors. But as we said, also, even if you could do this with molecules, I mean, there are a certain number of colors you can spectrally resolve in a microscope, but you will soon run out of colors. So you could do this maybe for 10 molecules. One way to do this is the notion of basically distinguishable. So what I want to show you is the example we had before. So we have an example with a lot of molecules in the square, and what you see here is just a blob, so undefined. But if you can identify each molecule separately, then of this identified molecule, you can easily determine the center. And of an individual molecule, identifying the center is easy because it's a circle, and finding the center of a circle, it's easy. And you can do this also very precisely. Even if your circle is pretty large, as we said, about a quarter of a micron, no? This shape of the point emitter, you can actually resolve the center of the circle with very high precision of the order of a few nanometers. So you can basically gain a factor of 100 in your resolving power, provided that you can find the center of individual molecules. And so if you do so, what you figure out is that you don't have just a blob here on the left, but you actually have, for example, 2 rows of molecules. And so you go basically from a diffraction limited world to a super resolved dimension where each molecule is replaced by basically a point which has the size of your localization precision. Okay, so the weaker the signal, the worse the localization precision. But if you have a good signal from your molecule, you can basically have a point which has basically an error of just 1 nanometer. And we're getting now closer to the higher resolution microscopy I was showing you earlier. Of course, how do you do this practically? How do you implement this practically? So one approach is to use molecules that basically can switch between a dark and a bright state. So these are called photo switches. For those who have a bit more experience in spectroscopy, basically you can trigger the chromophore of this fluorescent molecule to switch from an inactive state, where you see here that you don't have a band, so to say, to a state which can be actually excited, the open squares, where you actually have an excitation band. And basically, you can achieve this by light of a different wavelength. So for example, near UV light can be used to kick some of these fluorophores, which normally would be dark, into a state which is receptive to being excited by the classical excitation that, for example, you will use with a clean fluorescent protein. And so if you can do this, and you can couple this fact with the notion that a fluorophore, after that it has been emitting light for a certain amount of time, like the light bulb will go off because it will burn, then you can basically do patiently a cycle. So you could, for example, bring just a few of your fluorophore in this active state. Then you can visualize them. And so you will see something like this in your sample, just a few sparse fluorophore excited in a dark background. And then you will image them until they burn, they bleach. And then you will repeat with another set of molecules. And then for each set of molecules that you have in your image, you can localize them precisely. And so you store on the side the position of this molecule. And then in the end, you sum them all up. And the result of summing them all up leads you to a substantial increase in resolution, because now you basically are plotting just the centers of all your molecules. You got rid of this diffraction limited image, which was overlapping from each of the molecules. And so, for example, you can go from a visualization like this of a section of the endoplasmic reticulum, one of the organelles of the cell, to a visualization like this. I show this because this was from the first paper proving this concept in 2006. You can look at the skeleton of the cell, in this case, tubulin, which together with actin is one of the 2 key cytoskeletal proteins. And you can go from this type of visualization to this visualization, or even more strikingly, from this visualization, where you basically don't resolve really the individual fibers, to this visualization. Or you can look at nuclear pores on the nucleus of the cell. In a conventional microscope, they will look like this, so fuzzy and unresolvable. You remember my first attempt with the cell when I blew up the cytoskeleton. Or you can super resolve them with this sequential localization approach, and then you can see the individual nuclear pores. You can even resolve the individual subunits in the nuclear pore, which then lead you to a resolving power which is comparable to electron microscopy. Okay, so these images, which are really eye-catching, are meant to show you that the optical microscope, which somehow started unassumingly as a not very technologically innovative instrument compared to other microscopies which have become available over the last decades, can actually, with some clever combination of dyes and analysis schemes, can actually gives you resolving power, which is comparable to that of an electron microscope. Okay, what's the catch? The catch is that, of course, you have to do this sequential cycle of activations, localization, bleaching and repeat and repeat. And out of, sorry, I always skip the Nobel Prize winners, but this substantial achievement was recognized in 2014, so relatively shortly after, because the field somehow exploded in 2006 by awarding the Nobel Prize for chemistry, again for chemistry, not for physics, pointing to the fact that the probes and the developments in the analysis rather than the optics were the key here to these people. So I was saying, out of direct experience, so the catch is the fact that this type of approach to imaging is extremely slow. So these are the diffraction-limited images of some mitochondria near the plasma membrane of a cell, and this is the detail of a super-resolved mitochondria. You can see that here you don't see anything. Here you see that the mitochondrion actually curls around, and you can actually distinguish it as a specific organelle and feature. The question is that to acquire this image, which is not even, let's say, the state of the art, took about 3 hours. Now, the technology has clearly developed, and now one can do this much faster. But the bottom line is that the gain in spatial resolution here is achieved against, or let's say, at the expense of a strong reduction in speed. And so what you basically here can do is to look at dead samples which have been fixed and where molecules cannot move around, because if molecules start moving around as you're doing these cycles of excitation and localization, then your image will become all blurry again. Okay, so this was the gain in spatial resolution and to break the diffraction limit. Now, before we move to the applications, now I wanted to touch the other side of the story. So what if you want to study dynamical process? So pharmacology and biochemistry, which I was highlighting in the title, refer to clearly living organisms and very dynamic processes. So how can you now use a fluorescence microscope to touch these features? So we go back to our cell, and now we ask ourselves 3 questions. So the first question which I ask is, okay, so now this cell is alive. There are molecules, fluorescent molecules that form the actin cytoskeleton. I want to know how long a molecule on average sits in this position. Okay, so how do we do this? So now you are all expert in single molecules from what you saw from what I showed you before. So one could think that one could label individual molecules, and this has been done since the prehistory of, let's say, modern microscopies of the late 80s. One could label individual molecules, and at the time they were using a cam recorder, one could basically follow the particles, for example, attached to large gold nanoparticles. So your protein of interest could be attached to a large gold nanoparticle, and you could follow it as it moves. Or you could use a fluorescent ligand to label a receptor expressed on the surface of the cell. Then you will see individual receptor molecules moving, and since they are diluted enough, so they are sparse enough, you could still track and follow their trajectories as individual cells. So this would allow you to have information about the dynamics and how long molecules sit in a given region of space. However, this approach becomes unfeasible in 2 conditions. So if the particles become too crowded, and in cells things can be very, very crowded, or if particles are fast, because then to acquire these movies, you don't have enough resolution. It's like, so you would basically, you need some exposure time to actually see molecules precisely. But if during this exposure time your molecules move too fast, then you basically don't resolve anything anymore. And so what alternatives do we have? And this is the concept I wanted to now discuss with you. So one thing you could do is to basically define a very small excitation region with your fancy objective within the cell. So you try to make a spot of light as small as possible. So only things which will cross this spot of light, only fluorescence molecules which will cross this spot of light will actually be fluorescent. Otherwise, they will not be excited outside, and so they will be dark. So one of the main forces that drives things around in general, but also within a cell, is thermal agitation. So here we have molecules undergoing Brownian motion. This is a diagram from the original work of Ferran. Of course, the molecules here have nothing to do with proteins, but let's just use them as an example. So this is a molecule which is undergoing Brownian or random motion or thermal diffusion. And here we define this tight excitation volume, which I just showed you in the previous slide. So what will happen in this situation? What will happen is that your molecule, when they will cross the excitation volume, they will become fluorescent. So if you look from far away, what you will just see is a burst of light whenever a molecule enters your volume, and then things will go dark again as the molecule goes out. And as to my question about transit times and residence times of molecules, then you start to see where I'm going. Because of course you will have a signal in your excitation spot, which somehow is proportional to how many particles you have in your excitation volume. There could be more than one at once, but also your burst of light will also depend on the speed of the molecules, because a molecule which diffuses fast will cross rapidly through your excitation volume, so your burst of light will be short. If a molecule is slow, or made slow, or intrinsically slow, or the system is very viscous, whatever, then you will see a burst of light which is longer, because the molecule will stay in the excitation volume and therefore will emit fluorescence for a longer time. So a realistic trace, which you will get in a microscope, will more or less look like this now. So definitely more noisy, but you will definitely have burst in signal whenever your molecules cross your excitation volume. So one could say, okay, then I patiently measure the size or the duration of each of these bursts in order to determine what's the average transit time. And this can give me information about the process at hand or the diffusivity of my molecules, which are all important biochemical parameters. However, good luck if you do this by hand. So the question is, how can you do this more quantitatively and in a more clever way? So you can imagine to take this trace that you have collected and then basically slide it over itself. So I've shown here 2 sliding times. So you take your trace and then you start sliding it on itself. And then you just slide it a little bit and then you compare. Now, if your bursts have a certain average duration, which reflects the speed of the molecules. So if you slide it just a little bit for a duration which is shorter than the average size of the burst, then the peak will still overlap with peaks. So the trace will overlap well with itself. But as you slide farther and farther, you start to have a more de-overlapping or decorrelation. And then what happens is you will reach a point where a peak will correspond to a valley, like here, because you have slided for longer than the duration of the peak. So if you basically quantitate this degree of overlapping, what you can plot is something like this. So without being technical, this will be an autocorrelation function, but we could say that you have a high degree of overlapping here as you slide for a very short time. As soon as you slide your traces for longer than the average duration of your bursts, again, the transit time of the molecules, then your trace decorrelates or drops. And the position where this drops to half of its amplitude is actually the average transit time of your molecules, which in this case, this will be a small fluorophore in solution, gives you a transit time of the order of 10 microseconds. Of course, you need electronics, which is a bit sophisticated to go to these timescales, but this is just to give you the concept. So you can have information, average information about the dynamics of your molecules in your sample, even if you don't see the individual molecules, okay? The other question you can ask is, okay, now, another thing important in biochemistry or pharmacology, again, we'll go back to the title, if things come together or not. So 2 molecules will come together to do some work together, or a drug could bind a target to exert its action. So the question is, okay, now I want to know if 2 molecules in position 2 are by themselves or are in complexes. Okay, and then I want to show you this chart. Again, here, if there is time, I will maybe ask for a quick feedback. So you see these traces have the same average intensity, but they clearly look different. And can someone tell me why they are different? Or what makes them different? You can type in the chat, don't be shy. Also, this is a good way to check if people are still there. Well, yes, but which trace corresponds to the binding of 2 molecules? To Irene? Irene says the binding of 2 molecules. But which one will be the bound molecules? the blue, right? Oh, the second one, Irene, the second one will be in the red. Isabel says the blue, which will be the upper one. Okay, so the upper one, the blue one, is the dimer or the complex. The lower one is the monomer. So what's the difference here? The difference here is the jumps, or the variance of the trace. Because in the upper trace, so you still have the same average, because it means that the same number of fluorophores are inside your observation volume. But the problem is that when your fluorophores or your complex is, let's say in this case, a dimer, then when they move out of your observation volume due to this thermal agitation I was mentioning before, the jump is bigger because it's a jump of 2 fluorescent units as compared to the jump of 1, which you get when you have the monomers. So if you monitor the amplitude of the jumps, then you can make a statement on the oligomerization of your sample, which is another very important feature. And quantitatively, what you do is a histogram of your trace. Here, actually the color scale, just to make it confusing, is reversed. But basically, if the histogram of your signal, the amplitudes, Ray was saying the amplitude in the chat, if the histogram of the amplitude is fatter, then this means that your molecules are in a complex. Of course, you need a reference. If the amplitudes of your histogram the width of your histogram is smaller, so if your histogram is thin, then this means that you most likely are dealing with a monomer. Okay, so we can now do two things: measure the transit time of molecules, even if they're very crowded in a given spot, and we can see if they are together or not. Or this could apply also to ternary complexes or more complex situations. Okay, and then the last theoretical item before coming to the applications. So the last question was, how long does it take to go from position 1 to position 2? Or how can I monitor if my molecule is somehow traveling throughout the cell? Again, without being able to resolve individual molecules. And here we have to do a quick discussion on diffusion. So intuitively, diffusion is what you see here on the top. So you have a glass of water or a beaker, and then you have a dye, a drop of dye, which is left at time 0 here. Then it hits the surface, and then it starts to spread throughout your glass. Of course, these first 3 frames represent the out of equilibrium situation, where you start at time 0 with a high concentration in one point, and then the concentration becomes more and more homogeneous throughout. But once you reach this point, which is equilibrium, it's not that things don't diffuse anymore. Simply you don't see it because some molecules will go down, some other molecules will go up. So on average, everything still looks homogeneous, but there is still diffusion, so molecules moving around. So the question is, this will be what happens in the cell at equilibrium. So can I recover information on diffusions in a fluorescence microscope when I look at this situation? This situation will be easy, but this situation, you cannot just force the cell to pack all the actin in one corner and then say, now you're free to go spread throughout the cell, I see how fast you go. So we are always looking at this situation. So can I recover this information here? So these two circles here represent the 2 locations, one where you let the drop fall and one position farther away. So in more mathematical terms, what we are seeing here is that at time 0, when you drop the dye, the probability of finding the dye molecule in the red circle is very high. You have this curve, and it's very low away because there was no time to diffuse. As time progresses, then the probability of finding the dye elsewhere increases until at a very long time, the probability is homogeneous throughout. So can we do this type of analysis in the microscope? And this is what I want to show you. So this is an example. Here you see an image from a confocal microscope of the membrane of a cell. So it's green because there are receptors labeled with a green dye. You don't resolve really individual receptors because there are many and they are diffusing all over the place. So one thing you can do here is to basically take a movie. So you take many images like this and you take them very, very fast. And then you look at the profile of this movie and you obtain what you see here. This, let's call it a chemograph for those who know what they are. But basically you have a graph where you have time here, space here, which is the line, and then time here. So basically you have the line, then you measure the line again and again and again. And the gray values reflect the photons you're measuring here. So the intensity counts. So by eye, this looks like noise. But actually this noise encodes the information about the signal, the photons which are coming out of your fluorescence molecules which are exciting, and also the fact that they are moving. Because maybe a photon or a signal here, at this time here, could come, which came, let's say, from molecule 1. Molecule 1 could then diffuse and give you signal here. And is there any relation between the signal here and the signal there? So to make a long story short, one thing you can do is to basically go back to this idea of sliding things, no? The correlations I was showing you before. So you could slide this, what appears to be a noisy trace, in time and in space. And what you get after some analysis, and definitely we don't enter into the details, is a picture like this one. Now, this is called spatial temporal autocorrelation. It doesn't matter. So what you are basically seeing here is the drop of dye, which is broadening up in your glass, even though you are extracting this information from an equilibrium situation. So there is no dye here or no molecules in one point that then broaden up. But statistically, you can extract exactly this type of information. So at time 0, this axis is time and it's logarithmic. All your molecules, so your molecules start here. Over time, the probability that the molecule leaves this position because it's diffusing away increases and until you reach a homogeneous signal here. Okay, so the molecule will leave, and of course, the faster the molecule diffuse, the faster this kind of plume that you see here will open up. Because in water, or here is a 50% glycerin solution, just to give you the idea, this thing opens up rather fast. If you do it in a very viscous solution, honey or a high glycerin, then it will take a much longer time for the signal to decorrelate, if you were following my correlation analogy before, but also simply for the molecule to leave the center and diffuse away. Okay, I wanted to show you this just to give you the idea that you can do analysis on your data, even without seeing individual molecules, but you can reconstruct the average behavior with a very good temporal resolution, because here you see we are going below 1 millisecond in these traces. So why am I telling you all of this? So just keep in mind this graphical analogy. No? This is kind of a plume. It opens up fast if the molecule diffuses fast. It opens up slow, meaning the molecule sits for a long time in the same position before being able to diffuse away. Okay. So if you look at the rate of broadening of this object, what you are basically doing is exploring how rapidly molecules explore their surrounding. And this can be quantified by an analysis, which maybe many of you have already seen. It's called the mean square displacement. So you basically explore the area that a diffusing molecule covers as a function of time. And as time increases, the area explored increases. But if you look at shorter and shorter and shorter and shorter times, what you are also doing is also looking at smaller and smaller and smaller and smaller distances. And so the brain picking idea here is that if you do your analysis fast enough over time, this is a chart taken from the work of a very good friend, what you see is that the faster you go, also the smaller you look at. And this dashed line here represents the diffraction limit. So with this approach, even if we're not using any super resolving method per se, we can also look at the dynamics of molecules at spatial scales which are below the diffraction limit, this 1/4 of a micron I was mentioning at the beginning. Okay. And this is a non-entirely intuitive notion, but I think it's rather striking. Okay, and now in the last, let's say, 10 minutes, I will show you an application of all this discussion. So what all this is good for, no? Okay, so what about transit time, diffusion times? Can you do anything useful at all with this? So I will have to give you just a brief introduction about the biology, and then we move on to this data. So most cells, like the one I showed you before, expresses membrane receptors. These membrane receptors have the job of basically mediating extracellular signals and relaying them to the inside of the cell. A large family of these receptors are called G-protein coupled receptors. So if I scare you suddenly, what will happen is that adrenaline in your blood flow will increase, and it will bind adrenergic receptors in your heart, which will then increase your heartbeat. So this is a classical example. And this process happens because these adrenergic receptors bind adrenaline, told to an intermediate protein called the G protein, because they're called G protein coupled receptors, which then tolls to an enzyme called the adenylyl cyclase, which synthesizes cyclic adenosine monophosphate. So this cyclic adenosine monophosphate is what is called in cell biology as a second messenger, because basically the primary message was the signal outside, but then the end result is production of cyclic AMP in the cell. Okay, fine, then the cyclic AMP can bind proteins inside the cell and then they do some job. The interesting aspect here is that this signaling cascade is shared by a large number of different receptors, so which basically mediate very different signals. And then one naive question is, why do we have so many receptors if in the end result in the cell is always the same, meaning production of cyclic AMP? Also, and here take me on my words because we don't have the time to go into this, it's known, especially in a cardiac setting, that different receptors, which in the end both produce cyclic AMP inside the cell, stimulation of the different receptor leads to different effects. So the cell reacts differently, as if the cell knew which cyclic AMP is coming from which receptor, which is again the matter of distinguishing, like for the molecules. So it's as if cyclic AMP molecules were colored. How is this possible? This is a rather interesting paradox because it has to do basically with how the cells compartmentalize their signal. And this is even more paradoxical if we consider the fact that cyclic AMP is a pretty small molecule, which means that in water, it diffuses or in an aqueous solution like the cell, it diffuses very, very fast. So if you think of a cell, this is the cell seen by a physicist, an ellipse of 10 microns in a major axis. So what you see is that if you have a stimulus which produces cyclic AMP, let's say in one side, it takes a quarter of a second for the signal to basically go to equilibrium and fill the whole cell. So one could have said, well, maybe one receptor is here, the other receptor is here. So if you stimulate the receptor here, the signal stays local here. If you stimulate the receptor here, the signal will stay local here. This is most likely not the case. So there has to be another way, because this thing is too fast to be. And actually, the fact that this cyclic AMP was very fast was also observed microscopically by people. So what you have here is a sensor, doesn't matter how it works, which gives you a signal, becomes red as soon as cyclic AMP comes. And this is a very long cell protrusion, like an arm stretching out. And what you do is that you start cyclic AMP production here, and then you monitor how the red wave broadens or floods this arm. And you see that the front progresses, and by measuring how rapidly this front progresses, you can calculate the speed. 780 square micron per second is very, very fast. A protein in a cell has a speed or a diffusion coefficient of about 20 square micron per second. So it's more than an order of magnitude slower. So one would say, okay, so this molecule is very, very fast. But on the other hand, other people have seen, these are experiments done in Padova in 2002, people have seen that in certain cells, this is again a fluorescent reporter for the abundance of this cyclic AMP. The distribution is far from homogeneous. You see these striations in this neonatal cardiomyocyte, which tells you that in the green areas there is low amount of this cyclic AMP, and in these red areas there is a high amount. And now the question is, how can you catch this very fast molecule and make it confined? How do you manage to confine it spatially if it is so fast? So here I have this, so the only enzyme in the cell that can do the job is called the phosphodiesterase. Since in the paper is always displayed like this, I found the best analogy was the Pac-Man. So the phosphodiesterase chews up cyclic AMP molecules around and can create areas where you don't have any cyclic AMP. The problem is that the Pac-Man can be fast, but the phosphodiesterase as enzymes are very, very slow. And so this has led to a paradox because there is no way that the PDE Pac-Man can eat up all the cyclic AMP in order to create concentration gradients. So areas where there is no cyclic AMP, because it was all eaten up, it will be refilled immediately by the very fast cyclic AMP. And so to answer this question, in our group in Berlin, we thought that we could maybe try to look directly at cyclic AMP in fluorescence in a microscope. So we then built this molecule here, which is a cyclic AMP attached in the green square to a fluorescence molecule. This is fluorescein. It's a classical green fluorophore. This fluorescein has the special presence of two bonds here, which make it dark and cell permeable, so it can enter the cell, but once it's inside the cell, it becomes fluorescent. And we could verify that this hybrid molecule here still functions like the endogenous cyclic AMP because it can still bind its target. So you just check that basically it can still do the job in an in vitro setting that just cyclic AMP alone will do. And then we look at this under the microscope. You have the cells here in the fried egg view, the transmitted light I was showing you before. And then you have the fluorescence image. And the fluorescence image is definitely not very exciting. Here we just put a little bit of this cyclic AMP to be non-perturbative, just as a probe, not to disturb too much the system. So you basically see noise again. But as I showed you before, noise can be very informative. And this is exactly what was done. So we used the small fluctuations of these few cyclic AMP, fluorescent cyclic AMP molecules, which we inserted into the cell, to do this dynamic measurement. So this is in a different color scale, the plumes I was showing you before. So remember the dye diffusing in the glass of water. And what you see here is what happens if you feed some cells, a cell with this fluorescent cyclic AMP. So the pattern that you get is basically, if you remember what I was telling you, if you will put the dye not in water but in honey, the viscosity will be such that the thing will not diffuse anywhere. So what this graph is showing us is actually your fluorescent cyclic AMP that you added to the cell is not diffusing anywhere. So there is this long time tail is indicative of binding. So this cyclic AMP is stuck somewhere. And you say, wow, maybe the molecule gets stuck. It's nothing biological. But then we did, okay, so what happens if you force the cell to produce a lot of endogenous cyclic AMP, which will be non-fluorescent? So the cell, you stimulate this adenyl cyclase enzyme I was mentioning before to produce cyclic AMP, like a tsunami. And then this tsunami should basically, if your fluorescent cyclic AMP is bound to some specific sites in the cell, so proteins, then this wave of dark cyclic AMP should sweep it out. Then all the bound cyclic AMP will be the dark one, and your free cyclic AMP molecules, fluorescent molecules, will be again moving, and you should see the very fast diffusion of these molecules. Well, what we see is something reminiscent, because what happens when you do the stimulation is that you lose the bound component. So this is technically a displacement experiment, where you basically see that you could displace your probe, which means that it was bound to specific sites. So it was actually bound where also the native cyclic AMP will bind. And what does this relatively or apparently trivial observation mean? This means that in a cell, the number of binding sites for cyclic AMP outnumbers the actual cyclic AMP molecules. And this is important because then this leads to what is called buffer diffusion. So you can imagine like a pinball or un flipper in Italiano. Of course, if you have one ball, the ball will at some point fall into the traps. But then if you flood the pinball with balls, then all the traps will be occupied, and the few free balls that remain will be truly free, so they can roam the pinball without fear of being captured. So the point here is that the cell has, in normal conditions, so if you take a cell and you don't do anything to it, all the cyclic AMP is stuck. So whenever it jumps out of a trap, it will soon fall into another trap. And this also makes it effectively slow, so that this slow Pac-Man, the phosphodiesterases, can chew it up comfortably. As soon as the cell becomes more and more stimulated from the outside, from a stimulus from the outside, what happens is the number of cyclic AMP molecules increases. Then all these binding sites become saturated, and so the cyclic AMP molecules have the chance to start roaming freely. And then the phosphodiesterases, the Pac-Mans, cannot cope anymore as good. So what's the implication of this? Well, maybe this out of time constraints, I will skip. But so the implication of this is quite important, because in basal condition, the pigments can create an area around them which is depleted of cyclic AMP. This means that if a protein which needs cyclic AMP to function or to stop functioning is close by the phosphodiesterases pigments, this thing will be effectively protected, because any cyclic AMP molecule which comes will be basically always trapped, falling into this trap, meaning that the phosphodiesterase can catch it and actually create a protection region around the targets. However, as soon as you have a stimulus, and the stimulus could now be different depending on which receptor you're stimulating, then the cell is flooded. And so some of these depletion regions will be flooded, meaning your targets, depending also on the distance there from phosphodiesterases, will be bound by the cyclic AMP, whereas other targets, which are a little bit closer to the phosphodiesterases, could be still protected, being this depletion area. And this sets the stage for basically a very spatially localized, on the nanometer scale, compartmentalization of the cyclic AMP signaling. And this was new. Okay, so this was new, and I would say it's only thanks to the ability to finally visualize with a good fluorescence analog cyclic AMP dynamics within living cells that it was possible to address this, which was, I would say, really a truly open and highly debated question. So just to stick to the promise I gave in the abstract, I will just show 2 slides of 2 other applications. You're free to ask me at the end, or to contact me later if this had caught your attention. So of course you can use fluorescence microscopy to do way more things. So you can look at different type of cells. You see a heart cell here. And you can look at signal which comes from individual substructures. This is interesting because people have postulated that depending on where you stimulate the cells, you can have basically different cyclic AMP waves. And so this is an example where you can use microscopy to actually monitor this. Or you can look at other situations. For example, you can look at receptors which are important in cancer. Of course, you could follow them, you remember, as single molecules, but actually in cancer, it turns out that these receptors are actually overexpressed. So you actually have a situation more like this one, where the cell is flooded with these receptors. And then again, you have to rely on these statistical microscopy methods to make some quantitative statements about what these receptors do. And for example, if they aggregate and if this aggregation is functional for example, the proliferation and formation of metastases. Okay, so since I'm a bit over time compared to what I wanted to say, so the conclusion here is the following. So we have seen 2 ways to basically extract information at either increased spatial or temporal resolution from living cells using fluorescence microscopy. So one rely on breaking the diffraction limit the hard way and basically localizing molecules one by one and gaining spatial super resolution. The other one basically by just looking at statistics and going very fast. So the take-home message of this second aspect is that even if you cannot see single molecules, you can still follow them, or at least follow their ensemble behavior. And the application I showed you is telling you that an important signaling molecule like cyclic AMP actually is buffered in cells. And this allows the enzymes which degrade it, the phosphodiesterases, to create local gradients that can, on the nanometer scale, modulate the activation of downstream targets. And with this, I thank you for your attention, and I will be more than happy to take any question